You know, inside every single one of us, there's a fleet of trillions of microscopic, high-performance machines. They're working tirelessly every second of every day just to keep us alive. I'm talking about our red blood cells. And right at the heart of each one is a molecular engine of absolutely incredible sophistication, hemoglobin. So today, we're going to take a deep dive, a physiological journey really, starting from the molecular blueprint of this vital machinery and following it all the way to the systematic consequences when it all breaks down. Okay, this diagram right here pretty much lays out the whole system for us. Let's start on the left with the gas carrier vehicle. That's our red blood cell. Now, if you could peel back its surface, you'd see what's labeled here as the architectural framework. That's this amazing cytoskeletal lattice of spectrin and actin that gives the cell its structure and flexibility. Now, follow that line over to the right. We land at the molecular engine itself, hemoglobin. You can see it shown here in its two key functional states. The red one is oxyhemoglobin, all loaded up with oxygen, while the blue one is deoxyhemoglobin, which is ready to pick up more. And the whole point of all of this? Systemic gas exchange, delivering that precious oxygen to our tissues and hauling away carbon dioxide. It's a vital circuit that keeps the entire body running. So let's zoom in a little closer on this molecular engine. I mean, literally, all of gas exchange relies on this one molecule, hemoglobin. Adult hemoglobin, or HbA, is what we call a tetramer. That just means it's made of 4 polypeptide chains. You can see the 2 alpha chains in red and the 2 beta chains in blue. But here's where the magic really happens. What's nested inside each of those chains. That magnified view shows us the heme group. Each globin chain kind of cradles one of these groups in what's called a hydrophobic pocket. And right at the very center, you see the most critical component of all, a single atom of ferrous iron, Fe2+. That single atom, that is the precise spot where oxygen binds.
So how in the world is this complex engine actually built? Well, you can think of it like a biological assembly line. Heme synthesis kicks off inside the mitochondria, and it starts with 2 very basic ingredients, glycine and succinyl-CoA. Through a whole series of reactions, this ring structure of protoporphyrin gets built. Then comes the critical step. A ferrous iron atom, that Fe2+, is inserted right into the center of the ring, and boom, you've got heme. Finally, this brand new heme group is combined with a globin chain. This whole process happens 4 times, and then those 4 units come together to create the final functional hemoglobin tetramer. Oh, the red blood cell is this flexible biconcave disc about 8 micrometers across. But here's the crazy physical challenge it faces every single minute. It has to repeatedly deform and squeeze itself through capillaries that can be as narrow as 3.5 micrometers. That's less than half its own diameter. This requires an incredible amount of durability, especially when you think about the journey it takes. Over its 120-day lifespan, a single red blood cell travels about 480 kilometers, or 300 miles. To put that in perspective, that's roughly the distance from London to Paris. It's just an astonishing feat of biological engineering. So you might be wondering, how does this simple cell, with no nucleus or mitochondria, power such a long and difficult journey? Well, the answer is its internal power plant, the Embden-meyerhofpathway. It's a type of anaerobic glycolysis. As glucose comes in, this pathway gets to work, serving 3 absolutely critical functions. First, ATP generation. This provides the energy to maintain the cell's shape and keep it flexible. Second is NADH production, which is vital for an enzyme that keeps the iron hemoglobin in its functional state. And third, there's a little side path called the Luebering Rapoport shunt, and its job is to produce a molecule called 2,3-DPG. Now hang on to that thought, because we're going to see why that little molecule is so important in just a bit. Beyond just generating power, the red cell also needs a really robust defense system, and it has one. About 10% of its glucose is diverted into what's called the hexose monophosphate shunt, or hmpshunt. The key thing it produces is a molecule called NADPH. You can think of NADPH as a shield for the cell. It maintains a ready supply of reduced glutathione, and this glutathione protects hemoglobin and other proteins from oxidative damage. Now, if this defense system fails, like in a condition called G6PD deficiency, the cell becomes super vulnerable to destruction. The final piece of this vessel's amazing engineering is its hull, the cell membrane. On the surface, it's a standard lipid bilayer, but what gives the red cell its unique strength and flexibility is the complex protein lattice right underneath it. This lattice is made up mostly of spectrin and actin, and it forms this horizontal framework all across the inner surface. This framework is then anchored to proteins in the membrane, which provides vertical support. It's this intricate network that provides both the structural integrity to withstand all those shear forces and the flexibility to deform and squeeze through those tiny capillaries.
Let's take a look at the elegant, dynamic process of loading and unloading oxygen as the red blood cell makes its journey through the body. The entire function of hemoglobin, the whole thing, relies on its globin chains moving between two key states. On the left, in blue, we have the deoxy or tense state. See how in this shape the beta chains are kind of pulled apart? That creates a space for that 2,3-DPG molecule we talked about earlier. When 2,3-DPG slots in there, it stabilizes the molecule in a low oxygen affinity state. That's perfect for releasing oxygen into the tissues where it's needed. But then, look on the right in red. This is the oxy or relaxed state. As oxygen binds, the molecule literally tightens up, and it kicks out the 2,3-DPG. This relaxed state has a much higher affinity for oxygen, which makes it ideal for loading up in the lungs.
We can actually visualize this dynamic process on the system's dashboard, which is the oxygen-dissociation curve. Its famous S shape is a direct result of something called cooperativity. Basically, as one oxygen molecule binds, it makes it easier for the next one to bind, which causes that steep rise right in the middle. Now let's trace the normal operating range. The arterial point shows us that in the lungs, hemoglobin gets about 95% saturated with oxygen. As it travels out to the tissues, the venous point shows that saturation drops to around 70%. And that shaded area right between them, that's the functional delivery window. That's the oxygen that's effectively delivered to your body on each and every pass.
Now, this is where the system gets really smart. It can be calibrated based on the body's needs, and we see this as curve shifts. A right shift means that hemoglobin has a decreased affinity for oxygen, causing it to release O2 more readily in the tissues. This is triggered by signals that your body is working hard, things like high 2,3-DPG, high CO2, or a fever. On the flip side, a left shift means hemoglobin has an increased affinity. It tends to hang on to or hoard oxygen. The triggers for that are the exact opposite, low CO2 or hypothermia. This dynamic calibration ensures oxygen delivery is perfectly tuned to demand. So in our final section, we're going to look at the pathophysiology and the consequences of system failure, from a very specific fault in the engine all the way to the widespread condition of anemia. Let's start with a really specific engine failure called methemoglobinaemia. This happens when the iron in the heme group gets oxidized. Now remember, the functional heme group contains iron in the ferrous or Fe2+ state. That's the state that binds oxygen effectively. But in the dysfunctional state, that iron becomes ferric or Fe3+. It's just one extra positive charge, but that one tiny change changes everything. It cannot bind oxygen. This can be hereditary or it can be acquired through exposure to certain drugs and toxins. And the clinical result is something called cyanosis. That's a bluish discoloration of the skin from a lack of oxygenated blood. Okay, now for a much broader kind of system failure, anemia. It's defined as a reduction in hemoglobin concentration below the normal level for a person's age and sex. And you can see the specific thresholds for males, females, and newborns. But there's a really crucial point we have to consider here, and it's called the volume confounder. See, normally there's a good balance of fluid and cells in the blood. But in dehydration, for example, the low fluid levels can make the cell count seem falsely high. And on the other hand, in a state with high fluid levels, like pregnancy, the cells are diluted, which can give you a falsely low reading. So it's absolutely vital to consider a patient's fluid status when you're looking at these numbers. And the scale of this system failure, it's just immense. Anemia is a major global health issue. It affects roughly 1/3 of the entire world's population, with the highest prevalence among females and young children. You can see on the map that the areas with the greatest burden include large parts of Africa, India, and Southeast Asia. The causes vary by region, but they include major drivers like malaria, which destroys red cells, parasitic infections that cause blood loss, nutritional iron deficiency, which is very common, and genetic conditions like sickle cell disease. So, when the body experiences the reduced oxygen delivery that comes with anemia, it has this remarkable adaptation process. The trigger, of course, is the anemia or hypoxia itself. So in response, the body ramps up production of 2,3-DPG. This, as we know, causes a right shift of the oxygen curve, which forces hemoglobin to release oxygen more readily to the tissues. At the exact same time, a cardiovascular response kicks in, increasing heart rate and stroke volume to pump blood faster. And the clinical result of all this is that oxygen delivery at rest is often preserved, at least for a while. A slow onset of anemia allows these adaptations to work, but a rapid onset, like from major bleeding, causes severe symptoms very quickly. And here is where we can see the direct link between that underlying biology and what a patient actually feels and experiences.
The tissue hypoxia and the body's attempts to compensate lead directly to symptoms like fatigue and breathlessness. Vasoconstriction, which is the body shunting blood to vital organs, combines with the low hemoglobin to cause pallor or paleness, and that hyperdynamic circulation, with the heart working so much harder, can cause a bounding pulse. We can even see very specific physical signs like koilonychia, which are these spoon nails you see in iron deficiency, and even retinal hemorrhages in really severe cases.